Now we would do part 2 of the chapter, which is nutrition. How did you find the part 1? Let us know in the comment section below. Also, let us know what is your name and what part of the world are you watching this video from. I am Nidhi and I am from India. So, hi. Let's begin with nutrition now. When we walk or ride a bicycle, we are using up energy. Even when we are not doing any apparent activity, energy is needed to maintain a state of order in our body. We also need materials from outside in order to grow, develop, synthesize protein and other substances needed in the body. This source of energy and materials is the food that we eat. Now, how do living things get their food? The general requirement for energy and materials is common in all organisms, but it differs in different ways. Some organisms use simple food material obtained from inorganic resources in the form of carbon dioxide and water. These organisms, the autotrophs, include green plants and some bacteria. Other organisms utilize complex substances. These complex substances have to be broken down into simpler ones before they can be used for the upkeep and growth of the body. To achieve this, organisms use biocatalysts called enzymes. Thus, the heterotrophs' survival depends directly or indirectly on autotrophs. Heterotrophic organisms include animals and fungi. Let's first see autotrophic nutrition. Carbon and energy requirements of the autotrophic organism are fulfilled by photosynthesis. It is the process by which our autotrophs take in substances from the outside and convert them into stored forms of energy. This material is taken in the form of carbon dioxide and water, which is converted into carbohydrates in the presence of sunlight and chlorophyll. Carbohydrates are utilized for providing energy to the plant. We will study how this takes place in the next section. The carbohydrates, which are not used immediately, are stored in the form of starch, which serves as the internal energy reserve to be used as and when required by the plant. A somewhat similar situation is seen in us, where some of the energy derived from the food we eat is stored in the body in the form of glycogen. Let us now see what actually happens during the process of photosynthesis. The following events occur during this process: absorption of light energy by chlorophyll, conversion of light energy to chemical energy, and splitting of water molecules into hydrogen and oxygen, and reduction of carbon dioxide to carbohydrates. These steps need not take place one after the other immediately. For example, desert plants take up carbon dioxide at night and prepare an intermediate which is acted upon by the energy absorbed by the chlorophyll during the day. Now let's see some parts of the leaf. Midrib, vein, lamina or leaf blade are some of the parts of a leaf, and this is the cross section of a leaf. If you carefully observe a cross section of a leaf under the microscope, you will notice that some cells contain green dots. These green dots are cell organelles called chloroplasts, which contain chlorophyll. Now, let us tell you about an activity that we perform to demonstrate that chlorophyll is essential for photosynthesis. The activity goes like this. Take a potted plant with variegated leaves, which means leaves that exhibit different colors. For example, money plant, croton, or snake plant. Keep the plant in a dark room for 3 days so that all the starch gets used up. Now keep the plant in sunlight for about 6 hours. Pluck a leaf from the plant. Mark the green areas in it and trace them on a sheet of paper. Dip the leaf in boiling water for a few minutes. After this, immerse it in a beaker containing alcohol. Carefully place the above beaker in a water bath and heat till the alcohol begins to boil. Immerse it in a beaker containing alcohol. Carefully place the above beaker in a water bath and heat till the alcohol begins to boil. Can you guess what happens to the color of the leaf? Also, what is the color of the solution? You would notice that the color of the leaf disappears and it becomes colorless because chlorophyll dissolves in alcohol. The color of the solution becomes green. Now, dip the leaf in a dilute solution of iodine for a few minutes. Take out the leaf and rinse off the iodine solution. Now, if we observe the color of the leaf and compare this with the tracing of the leaf done in the beginning, we would find that the green part of the leaf turns blue-black and the iodine solution will be orange-brown in color. Now, what can we conclude about the presence of starch in various areas of the leaf? The presence of starch indicates that photosynthesis occurs only in the green part of the plant and chlorophyll plays an important role in photosynthesis. Now, let us study how the plant contains carbon dioxide. How does it obtain carbon dioxide? In class 9, we had talked about stomata, which are tiny pores present on the surface of the leaves. Massive amounts of gaseous exchange takes place in the leaves through these pores for the purpose of photosynthesis. But it is important to note here that exchange of gases occurs across the surface of stems, roots, and leaves as well. Since large amounts of water can also be lost through these stomata, the plant closes these pores when it does not need carbon dioxide for photosynthesis. The opening and closing of the pore is a function of the guard cells. The guard cells swell when water flows into them, causing the stomatal pore to open. Similarly, the pore closes if the guard cells shrink. Pretty interesting, isn't it? Now, let me tell you about one more activity. Take two healthy potted plants which are nearly the same size. Keep them in a dark room for 3 days. Now place each plant on a separate glass plate. Place a watch glass containing potassium hydroxide by the side of one of the plants. The potassium hydroxide is used to absorb carbon dioxide. Cover both plants with separate bell jars. Gel bars. Bell jars. Use Vaseline to seal the bottom of the jars to the glass plates so that the setup is airtight. Keep the plants in sunlight for about 2 hours. Pluck a leaf from each plant and check for the presence of starch as done in the previous activity. What do you think? Do both the leaves show the presence of the same amount of starch? Well, the answer is no. Both the leaves do not show the presence of the same amount of starch because in photosynthesis, starch is produced using sunlight, chlorophyll, and carbon dioxide. In the first setup, availability of carbon dioxide will be less for making starch by the plant leaves as potassium hydroxide absorbs the carbon dioxide. So, From this activity, we can conclude that amount of carbon dioxide essentially affects the process and outcome of photosynthesis. Based on the two activities performed above, can we design an experiment to demonstrate that sunlight is essential for photosynthesis? Well, so far we have talked about how autotrophs meet their energy requirements, but they also need other raw materials for building their body. Water used in photosynthesis is taken up from the soil by the roots in terrestrial plants. Other materials like nitrogen, phosphorus, iron, magnesium, these are taken up from the soil. Nitrogen is an essential element used in the synthesis of proteins and other compounds. This is taken up in the form of inorganic nitrates or nitrites, or it is taken up as organic compounds which have been prepared for bacteria from the atmospheric nitrogen. Next is heterotrophic nutrition. Each organism is adapted to its environment. The form of nutrition differs depending on the type and availability of food material as well as how it is obtained by the organism. For example, whether the food source is stationary or mobile would allow for differences in how the food is accessed and what is the nutritive apparatus used by a cow and a lion. There is a range of strategies by which the food is taken in and used by the organism. Some organisms break down the food material outside the body and then absorb it. Examples are fungi like bread molds, yeast, and mushrooms. Others take in whole material and break it down inside their bodies. What can be taken in and broken down depends on the body design and functioning. Some other organisms derive nutrition from plants or animals without killing them. This parasitic nutritive strategy is used by a wide variety of organisms like Cascata or Amarbale, ticks, lice, leeches, and tapeworms. Now, the next important question is, how do organisms obtain their nutrition? Since the food and the way it is obtained differ, the digestive system is different in various organisms. In single celled organisms, the food may be taken in by the entire surface. But as the complexity of the organism increases, different parts become specialized to perform different functions. For example, amoeba. It takes in food using temporary finger like extensions of the cell surface which fuse over the food particle, forming a food vacuole. Inside the food vacuole, complex substances are broken down into simpler ones, which then diffuse into the cytoplasm. The remaining undigested material is moved to the surface of the cell and thrown out. In paramecium, which is also a unicellular organism, the cell has a definite shape and food is taken in at a specific spot. Food is moved to this spot by the movement of cilia, which cover the entire surface of the cell. Next is nutrition in human beings. The alimentary canal is basically a long tube extending from the mouth to the anus. Here we can see that the tube has different parts. Various regions are specialized to perform different functions. What happens to the food once it enters our body? We shall discuss this process here. Let me first tell you about an activity. Take 1 ml starch solution, 1%, in two test tubes A and B. Add 1 ml saliva to the test tube A and leave both test tubes undisturbed for 20 to 30 minutes. Now add a few drops of dilute iodine solution to the test tubes. What do you think, in which test tube would you observe a color change? Color change will be observed in test tube B where saliva is not added. Now, what does this indicate about the presence or absence of starch in the two test tubes? Well, it indicates that the iodine changes the color of starch. If starch is present, then blue-black color is formed. And lastly, what does this tell us about the action of saliva on starch? Saliva consists of salivary amylase, which acts on the starch and convert it into glucose. We eat various types of food which has to pass through the same digestive tract. Naturally, the food has to be processed to generate particles which are small and of the same texture. This is achieved by crushing the food with our teeth. Since the lining of the canal is soft, the food is also wetted to make its passage smooth. When we eat something we like, our mouth waters. This is actually not only water but a fluid called saliva secreted by the salivary glands. Another aspect of the food we ingest is its complex nature. If it is to be absorbed from the alimentary canal, it has to be broken into smaller molecules. This is done with the help of biological catalysts called enzymes. The saliva contains an enzyme called salivary amylase that breaks down starch, which is a complex molecule, to give simple sugar. The food is mixed thoroughly with saliva and moved around the mouth while chewing by the muscular tongue. It is necessary to move the food in a regulated manner along the digestive tube so that it can be processed properly in each part. The lining of canal has muscles that contract rhythmically in order to push the food forward. These peristaltic movements occur along the gut. From the mouth, the food is taken to the stomach through the food pipe or esophagus. The stomach is a large organ which expands when food enters it. The muscular walls of the stomach help in mixing the food thoroughly with the more digestive juices. The digestion in stomach is taken care of by the gastric glands present in the wall of the stomach. These release hydrochloric acid, a protein digesting enzyme called pepsin, and mucus. The hydrochloric acid creates an acidic medium, which facilitates the action of the enzyme pepsin. What other function do you think is served by the acid? The mucus protects the inner lining of the stomach from the action of the acid under normal conditions. We have often heard adults complaining about acidity. Can this be related to what has been discussed now? Let us know your thoughts in the comment section below. The exit of food from the stomach is regulated by a sphincter muscle, which releases it into small amounts into the small intestine. From the stomach, the food now enters the small intestine. This is the longest part of the alimentary canal, which is fitted into a compact space because of extensive coiling. The length of the small intestine differs in various animals, depending on the food that they eat. Herbivores eating grass need a longer small intestine to allow the cellulose to be digested. Meat is easier to digest, hence, carnivores like tigers have a shorter small intestine. The small intestine is the site of the complete digestion of carbohydrates, proteins, and fats. It receives the secretion of the liver and pancreas for this purpose. The food coming from the stomach is acidic and has to be made alkaline for the pancreatic enzymes to act. Bile juice from the liver accomplishes this in addition to acting on fats. Fats are present in the intestine in the form of large globules, which makes it difficult for enzymes to act on them. Bile salts break them down into smaller globules, increasing the efficiency of the enzyme action. This is similar to the emulsifying action of soaps on dirt. The pancreas secretes pancreatic juice, which contains enzymes like trypsin for digesting proteins and lipase for breaking down emulsified fats. The walls of the small intestine contain glands which secrete intestinal juice. The enzymes present in it finally convert the proteins to amino acids, complex carbohydrates into glucose, and fats into fatty acids and glycerol. Digested food is taken up by the walls of the intestine. The inner lining of the small intestine has numerous finger like projections called villi, which increase the surface area for absorption. The villi are richly supplied with blood vessels, which take the absorbed food to each and every cell of the body, where it is utilized for obtaining energy, for building up new tissues, and for the repair of old tissues. The unabsorbed food is sent into the large intestine, where its wall absorb more water from this material. This, the rest of the material is removed from the body via the anus. The exit of the waste material is regulated by anal sphincter. So this is how the digestive system functions in humans. Let's talk about something different now that we should know, and that is dental caries. Dental caries or tooth decay causes gradual softening of the enamel and dentine. It begins when bacteria acting on the sugars produce acids that softens or demineralizes the enamel. Masses of bacterial cells together with food particles stick to the teeth to form dental plaque. Saliva cannot reach the tooth surface to neutralize the acid as plaque covers the teeth. Brushing the teeth after eating removes the plaque before the bacteria produce acids. If untreated, microorganisms may invade the pulp, causing inflammation and infection. So, do take care of your teeth, brush them regularly, and rinse your mouth after every meal. So, this was about nutrition part 2 of this chapter. Let's try to answer some questions before moving on to the next part. First question is, what are the differences between autotrophic nutrition and heterotrophic nutrition? Autotrophs are organisms that prepare their own food through the process of photosynthesis, whereas heterotrophs are organisms that cannot prepare their own food and depend upon autotrophs for nutrition. Autotrophs are producers in the food chain, for example, plants, whereas heterotrophs are consumers in the food chain, for example, us, animals. Moving on to the next question. Where do plants get each of the raw materials required for photosynthesis? Carbon dioxide. Plants obtain carbon dioxide from the atmosphere via stomata. Water. Plants take water from the earth and transmit it to their leaves via their roots. Now, sunlight is the only source of light that the chlorophyll and other green elements of the plant absorb. What is the role of the acid in our stomach? The hydrochloric acid in the gastric juice breaks down the food and the digestive enzymes split up the proteins. The acidic gastric juice also kills bacteria. The mucus covers the stomach wall with a protective coating. Next question is, what is the role of the acid in our stomach? Digestive enzymes such as amylase, lipase, pepsin, trypsin, etc. help in the chemical digestion of food by breaking down complex food particles into simpler ones. So, sorry, the question was, what is the function of digestive enzymes? So, digestive enzymes such as amylase, lipase, pepsin, trypsin, they all help in the chemical digestion of food by breaking down the complex food particles into simpler ones. These simple molecules can be easily absorbed by the blood and is thus transported to all the cells in the body. And the last question from this part is, how is the small intestine designed to absorb digested food? The small intestine has millions of tiny finger like projections called villi. These villi increase the surface area for efficient food absorption. Within these villi, many blood vessels are present that absorb the digested food and carry it to the bloodstream. And hey, congratulations! We just completed part 2, nutrition. If you like the video, do give us a thumbs up, subscribe to the channel, and share it with your friends and family. Have a wonderful day. Keep watching I Say Kids, keep loving us, keep writing to us, and keep sharing our videos. Have a lovely day. Bye bye.